Our topic today is microscopes. So we're going to talk about two different types of microscopes. It's over at the front. We're going to talk about two different types of microscopes that exist and what some of the advantages and disadvantages of each type of microscope is. So this first slide is about what we call light microscopes. So I might actually write that first and then write the two types of light microscopes that exist. A light microscope, which can sometimes also be called an optical microscope, is one that uses lenses to magnify the light that is reflected off an object. And then there are two varieties of light microscopes that use lenses that I would want you to be aware of. So I'll just pause here for a second. So we'll talk about light microscopes. The reason I'm pointing this out, and you might say to yourself, what other kind of microscope is there other than one that I look in? There are other kinds of microscopes that use something other than just visible light to get an image of a small object that we're trying to magnify. In the light microscope category, the thing that's really key is that it uses lenses. And I'm pointing that out because the way that we magnify is not going to be lenses in the second type. A simple microscope uses one lens. So if you think of what a magnifying glass looks like, that is a simple microscope. It's one lens, and if you ever had your junior science explorer kit when you were a kid, you got a magnifying glass so you could look at bugs. That was a simple microscope. The idea is that a lens helps magnify the light that comes off of the object. I won't get into the physics of why that actually works, but just so that you're clear, a lens bends light. So when you were in grade 8, you learned about lenses at some point and how they do refraction. Refraction is when light comes in on a straight line and then comes out of the lens on a bent line. That's the way that a lens can make an image look bigger. A compound microscope is the one that we will focus on because it's the one that we use in the lab. A compound microscope uses at least two lenses. There would be microscopes that have more than two, but the ones that we have in the lab have two. The eyepiece and the objective lens. On your picture, that's number 1 for the eyepiece lens and number 10 for the objective. And the idea is that two lenses together can be more powerful than only one lens. So that's what a light microscope is. It's something that uses lenses to magnify light. Now, sometimes this is useful, and sometimes perhaps it's not. And so these 2 points here are telling you maybe when I would and when I wouldn't want to use a light microscope. So you could expect that when we do our next quest, I would give you a situation, I would tell you what I'm trying to do, and I would ask you, which type of microscope should I use? If I am trying to look at something in an inexpensive way, or very specifically, I'm trying to look at something that is alive, I probably want to use a light microscope. So light microscopes do not cost a lot of money, and they're simple. They're simple to use, simple to set up. But those are sort of technical things that aren't really scientific. The scientific advantage of a light microscope is that the specimen, so the cell or the organism I'm trying to look at, can still be alive when I look at it. And that's a big deal a lot of the time. Some of the disadvantages of a light microscope is that there is a limit. Generally, they can only magnify up to about 4,000 times. That would be based on the ones that we have in the lab. But the idea is that there's a limit. They can only magnify so much. And the reason is, after a certain point, resolution decreases if I try to magnify any further. Now we'll talk about what resolution is, but resolution is the clarity. So how sharp the picture is. Do the edges look fuzzy, or can I clearly distinguish all of the objects that I'm looking at? So before we talk about the other kind of microscope, I would just like to recap with you guys what the functions of all of the parts of the microscope are. I asked you the day before, when we were talking about microscopes, to use the handout that I gave you. It's called Student Reference 8, has a picture of a microscope, and it tells you instructions for using the microscope. And I just want to make sure everyone is clear on what the function of each of the parts of the microscope is. Before I do that, does anyone have any questions about the description or the advantages or disadvantages of light microscopes? So, this is the picture that I handed out to you all. If it's not in front of you, maybe you'd like to take the picture out so that you could make sure you have the correct name and that you have the function of each of the parts of the microscope on there. That way, when you look back at that sheet, it becomes a really useful study tool. So I'll just make my screen a little bit bigger here. So on your picture of a microscope, we'll just kind of go in order. Part number 1 is called the eyepiece. Now the eyepiece, or the ocular lens, as well as part number 10, the objective lens. So if I write over here, number 1 and number 10 magnify the image. So those are your two lenses. Those are what are actually making the image appear larger. Part number 2 is the arm, 6 is the base. Number 2 and number 6 both are there for support. Now when we are in the lab, those are the two pieces that you are supposed to hold on to on the microscope when you're carrying it around, because structurally they support the microscope. Part number 3 is the stage, and I'm just going to point out part number 9. They are the stage clips. Part 3 and 9 hold your slide. So the stage is the flat surface on which you are putting your slide. The clips are what hold it there so that it doesn't move around while you're trying to look at it. Part 4 and part 5 are both things that you can turn. And so this is a place where they do kind of the same thing, but I would really like for you to know the difference between when you're supposed to use one versus the other. So on the handout that I gave you, it tells you that when you're looking at things in the microscope, the first thing you're supposed to do after you've gotten your slide in place is to use the larger one of the knobs. I'm just trying to find the page here. So it says in step number 8 to slowly turn the coarse adjustment knob. So this is coarse adjustment. Both of the knobs are going to help you bring your specimen into focus, but the coarse adjustment knob makes a bigger change to the focus. What it's actually doing is making the stage go up or down by like a visible, distinct amount. You should always use the coarse adjustment knob first, just so that your specimen is like in the field of view. The reason it says carefully is because if you turn the coarse adjustment knob too much, you will hit the lens with the stage, and you don't want to do that because you could break the lens. Once you have your specimen in your field of view, and you can see it, even though it might not be really clear, then you use the fine adjustment knob, and it is for focusing. So the coarse adjustment knob is what you use when you're just trying to find your specimen and make it show up in your field of view. Once you can see it, even though it's not clear, that's when you use the fine adjustment knob to really bring things into focus. Number 7 is the light, or sometimes a mirror, depending on what type of microscope. And number 8 is called the diaphragm. It's actually spelled diaphragm, but you don't say half of the letters. It's just like the diaphragm that you have as a muscle in your body, in that it controls the amount of something. The diaphragm here controls the amount of light that is getting through onto the slide. So the function of part number 8 is to control the amount of light. If we keep going around the top, 11 is the revolving nose piece. It's the thing that allows you to select which objective lens you're using. And number 12 is called the body, or the body tube. And it's there so that there's a space between the two lenses. Having space between the two lenses allows the image to be magnified properly and for it to show up in the right direction when you look at it. Does anyone have any questions about the parts or the function of the parts of a microscope? So the next thing that we'll talk about then are electron microscopes. So we'll just make this, well, that's a good size. So electron microscopes are the other kind of microscope we'll discuss. These are not the only two kinds that exist in the world. They're just the kinds that we'll focus on. An electron microscope uses electrons, not light, electrons, to create an image that you see on a monitor. So notice, I'm not shining light on something, and I'm not actually looking at it with my own eye. Electrons are making some sort of picture that a monitor is going to set up for me, and that's what I'm going to look at. So on this page, anytime I use the abbreviation EM, I mean an electron microscope. So an electron microscope uses electrons to create an image seen on a monitor. And then there'll be two kinds. So the first kind of electron microscope is a scanning electron microscope, which I've abbreviated scanning EM. Now, if you think about what the word scan means, a scan is when you look at the surface of something, or when you're trying to get a general idea of the outside of something. So a scanning electron microscope is one where we bounce electrons off the surface. And so the idea is the surface of whatever I'm trying to look at is coated with a thin film of something like gold. And I'm going to get a 3D image of the exterior of the object. So I'll pause. If I cover something, the surface of something, some cell, with a thin film of gold, do you think that that thing is still alive? It's not. So because of this, I am only allowed to look at dead tissue. In fact, when I started watching Game of Thrones, more or less because my husband peer pressured me into it, claiming it's so good, how can you not watch it? There was an episode, I don't know if any of you have seen this show, where they melt a guy's crown onto his head. And what I thought of was this. I'm like, oh, look at that. He's covered with a thin film of gold, and he's not alive anymore. He is most definitely dead. And so I thought about that little scene and a scanning electron microscope. The reason then that you would want to do this, even though the cell has to be dead, is because of the really awesome 3D image that you get of the outside. We're only looking at the outside, but we get really high resolution and really good magnification. And so I'm just going to bring this little picture up here for you to look at for a second. That's a scanning electron microscope picture showing you the gills of a fish. And that's a really, really clear picture. If you look, that's great 3D representation. We're very close, so it's very high magnification. But we're only looking at the outside. I can't see what the inside of that structure looks like, but sometimes the inside is not what I'm interested in. And so it all depends what your goal is when you're using the microscope. Now the second kind is a transmission electron microscope. And so think about what the word transmission means. I'm sending something through. And so the word through here is one that I like to make jump out. In a transmission electron microscope, electrons are shot through the specimen. And as they're shot through, they scatter, and they scatter because they hit things. So the idea would be if there was no structure to hit, the electron would keep going straight. But as soon as it hits something, it scatters. And that's how the computer knows that a structure was there. The scattering of the electrons is recorded, and then we get an image. This type of microscope lets us view the internal structure of cells, again, with a really high magnification and really high resolution. So I'll just pause here. So just like with a scanning electron microscope, a transmission electron microscope, because it is shooting electrons through things, kind of requires that the specimen be dead. And so at the bottom, I've listed some advantages and disadvantages of using electron microscopes. And they're kind of the opposite of what the advantages and disadvantages were for light microscopes. So for example, in an electron microscope, I get awesome magnification and resolution. Whereas in a light microscope, if I tried to have higher magnification, I got really terrible resolution. The disadvantages are kind of the opposite of the advantages for light microscope. It's expensive, it's hard to use, and you can only look at dead tissue. So the idea would be that an electron microscope is useful when you need a really high magnification or high resolution picture of sometimes the exterior of the object or sometimes the internal structure. Would anyone like to ask any questions about the two types of microscopes? So what we'll do next then is start talking about the properties of microscopes that you will observe or use when we do our lab. You heard Mr. Van Erf come in here. We're going to go to the lab on Tuesday so that you guys can start using microscopes. Is there anybody who is still writing? I see a few people, so I'll just pause. All right, so I think we're ready to move ahead. We are going to talk about some of the properties of light microscopes. Some of them are just descriptions. Some of them are calculations that we'll do. But all of these are things that we're going to observe or things that we're going to make use of when we go to the lab next week. And so the first thing that I'd like to mention is this idea of inversion. So when light passes through a lens, you may or may not be aware of this, but it is inverted. And so, for example, you have a lens in your eye. What you are seeing in your brain, like what you are seeing as the picture, is what your brain is interpreting. If you were to actually look at the picture that came through the lens on your eye, it's upside down. And so inversion is that when light passes through a lens, up is down and left is right. So what does that mean for you? That means that if you would like to move the specimen that you're looking at in your field of view, and it looks like you want to move it to the left, you actually want to push it to the right. So this is important for moving your specimen. Specifically, which direction are we going to move the stage? Because by moving the stage, we can adjust the position of our specimen. And the idea is that when you look through the microscope, whatever direction you're actually pushing it in, it'll look like it's moving the other way. And it just takes a little while to get used to that. So I'm just showing you an example here of the letter E, how it looks upside down. And I picked the letter E because I'm going to ask you to look at a letter E under the microscope and to note some things about what you can and can't see. The next property is called resolution. And so the actual definition of resolution is the capacity to distinguish two close structures, one from the other. If I had to sum it up, I would say it's the clarity. But technically, resolution is the capacity to distinguish two close structures, one from the other. That means that we measure distance to give a number to resolution. So for example, I might tell you that a particular microscope has a resolution of 20 nanometers. Does anybody know what a nanometer is? So in the metric system, the beginning of the word tells you which power of 10 it represents. You all know what a meter is, I'm sure, even a centimeter or a millimeter. A micrometer is what comes next, and then a nanometer. So a nanometer is 10 to the power of -9 meters. So it's 1,000,000 times smaller than a meter. It's 1,000 times smaller than a micrometer. But that's a nanometer. What we would be saying then is that microscope could make you clearly see two objects that were that close together. So that's usually what we're measuring. The closer together two objects are, the better the resolution. So if magnification increases, something to take note of is that you are looking at a smaller part of the object and less light is focused, resulting in a less clear image. That sentence explains why, as magnification goes up, resolution goes down. So the first calculation that you'll have to do is a relatively easy one. And I find that the reason people make mistakes here isn't because they don't know how to multiply. It's because they forget, am I supposed to add or divide or multiply? Which process am I actually doing? So the idea is that to calculate magnification, we need to consider the ways that the light is being magnified. The light is magnified as it goes through two lenses, the objective lens first, then the ocular lens, then it enters your eye. So actually, technically, it's going through three lenses because you have a lens in your eye, but we'll just focus on the microscope. Our microscopes that we have at school have one ocular that magnifies 10 times. And most of them only have 3 objectives. A few of them have 4. But the 3 objective lenses magnify 4, 10, and 40 times. So that little X represents times. So to calculate magnification, all you need to do is multiply the magnification of the ocular times the magnification of the objective. So as an example, I could ask you really 2 kinds of questions. I could ask you a simple calculation. I could ask you, after having told you this information, what's the magnification possible on the microscopes in our biology lab? And you would say, well, it could be 4 times 10, so 40 times. It could be 10 times 10, so 100 times. Or it could be 10 times 40, so 400 times. So to calculate magnification, you need to know what's the value of the ocular, what's the value of the objective. We would call this low, medium, and high power. Now that would not be the only way that I would ask you about this. I might ask you, not just by telling you in the question what the value was, I might ask you a question by showing you a picture of the microscope that we're talking about and having you figure out the values. So the other way I could ask this would be from a picture. So if I was going to make a picture, here are the things that you would want to look for. You would want to look on the eyepiece, usually right here, and it'll say some number and then an X. That would be the magnification of the eyepiece. Oh, look at that, big sister. What do you say to her for that? The next thing that you want to look for is on the revolving nose piece, you'll have your objective lenses. There would be a medium one, a long one, a short one. There might even be a 4th one, but usually 3 is the standard. The shortest one is your low power. The medium-sized one is medium power. And the longest one is high power. And so you would have to recognize by size which one was which. Each of the lenses will have a number and then an X. So let's say, just for fun, that low said 6 times, high said 72 times, and medium said 11 times. I could ask you, What's the low power magnification on this microscope? And so you would say on low, 10 times 6 is 60 times. Notice how I wasn't just writing 60, I was writing 60 times, because that's the unit that I would give for magnification. So I'm just going to pause here.